Hello everyone, welcome to Istanbul again. My name is Gun Kaynar and I'm a master's student at Bilkent University where I am fortunate to work with Ercument Cicek. So I will present our recent work on developing a pathway-informed deep learning model for the survival analysis of gliomas. To give some background, gliomas are the most common type of brain tumor, accounting for nearly 60% of all primary brain tumors. And according to the Central Brain Tumor Registry of United States, the annual incident rate is 6 per 100,000 persons, which is very big. So despite the recent advancement in treatment options, these glioma patients still face a poor prognosis with a median survival of 18 months, unfortunately. So therefore, we see an urgent need for developing a deep learning model for prognosis prediction for these patients. So here is a healthy brain image followed by benign and malignant glioma images. So these tumors are very heterogeneous and highly complex with different histopathological, molecular, and genetical characteristics. These characteristics include different cell types, such as astrocytes or oligodendrocytes, different grades ranging from 1 to 4, and different malignancy levels, either benign or aggressive. So we can say that maximum resection of the primary tumor has a direct influence on the survival of patients. However, given the complexity and heterogeneity, making this decision during the surgery may be very challenging. So this is where our work comes in. We want to develop a personalized decision-making feedback mechanism that can help surgeons make more informed decisions during surgery. So in this work, we utilized a promising technology for tissue analysis. It is called HRMAS NMR spectroscopy. So it stands for High Resolution Magic Angle Spinning Nuclear Magnetic Resonance. Okay, thank you. So this technique is very novel. So it has several advantages over the traditional tissue analysis methods. The first one is it allows for the analysis of small samples of unprocessed tissue specimens. So as there is no need for processing, it allows for faster sample preparation. And it's non-destructive, meaning that the tissue specimen remains intact during the whole analysis. So our prior work before relied only on feedback mechanism during, our feedback mechanism during surgery relied only on tumor malignancy prediction. Nevertheless, merely relying on only malignancy prediction may be insufficient because we have some cases of malignant glioma with a prolonged survival for the patients. Similarly, rapid progression and low survival can happen in some types of benign glioma too. So we want to integrate the survival prediction information with tumor malignancy prediction information here. So for an online mechanism of this kind, we need a robust, accurate, and interpretable model. Interpretable because surgeons will use it. However, our data is limited to under 400 samples. So one way to achieve deep learning in small data regimes is by integrating prior knowledge into neural networks. And this approach has shown promise in overcoming overfitting because it involves fewer connections between the layers. So these are the prior work done on biologically informed neural network architecture. On left, you see a biologically informed neural network for prostate cancer discovery. So in this model, input features such as mutations, copy number variations, methylation profiles, and gene expressions, they are connected to the corresponding genes. And these genes, as represented by neurons, are connected to pathways they are involved in, followed by biological processes and the outcome. On right, you see a phenotype prediction model. It is again biologically informed because input features in this one is SNPs. They are connected to genes they reside in. and these genes are connected to local and global pathways they are involved. So these works demonstrate the potential of integrating biological knowledge and also provide a context for our own work, which is a completely different problem. Okay, this is the proposed pipeline for our model. PyDeal, we named it PyDeal. It means pathway-informed deep learning model, basically. So this feedback mechanism involves several steps. The first, surgeon removes the primary brain tissue from the glioma patient's brain and prepares specimens from this resected tissue. These specimens are then sent to HRMS NMR spectroscopy. After the preprocessing of the output from the HRMS NMR spectroscopy, we use our metabolite-specific quantification models to quantify the 37 metabolites of interest. So our model, PyDeal, then transforms this information on concentration of metabolites to survival prediction. This way, it allows for the efficient and accurate feedback to the surgeon during surgery. So this is moving on to the data set. This QR code directs to the Zerado repository where we published our data set. You can save it if you want. So we utilized HRMAS NMR dataset, which included 384 samples in total. Out of these, 299 were aggressive and 85 were benign tumors. So these datasets are collected in university hospitals of Strasbourg. The surgeons collected for us, our collaborators. So you can see the age, time to loss control, and time to disease distributions of patients in our data set. In this one, we show the number of samples for each histopathological type of gliomas. As you can see, it's very diverse. So I will quickly guide you through our pre-processing pipeline. This is the raw output or FID signal from HRMAS NMR spectroscopy. The first 70 elements of this spectrum are instrument specific, so it has no information on either sample or patient, so we removed those. After that, we transformed our spectrum from time domain to frequency domain using Fourier transform, and followed by phase correction. After that, we quantified 37 metabolites of interest using our automated metabolite quantification pipeline from prior work. So in this slide, I will give the problem formulation we used for our study. So for each sample, we have three kinds of information: metabolite concentration vector, survival event indicator, which means patient has died or not, and time to event variable. So we use Cox's proportional hazards model, which is a widely used statistical model for survival analysis. And this proportional hazards function factorizes to a time-dependent baseline hazard function, H0 of t, and a sample-dependent risk function, H of m in our case. So the formulation given by this equation allows us to model the influence of metabolite concentration vector on the prognosis or survival of the patient, while also considering the baseline hazards. So we trained survival analysis models to learn a mapping function f that takes in 37-dimensional metabolite concentration vector and outputs the hazard rate for each patient. So in other words, we want to predict the likelihood of an event occurring for a given patient based on their metabolic profile. So now let's take a closer look at PyDeal's architecture. So as you can see, metabolite concentration are given as input, and the neurons in the first hidden layer represent pathways. We have 138 pathways in our case because of the number of metabolites. So we only connect these metabolites to the pathways they are involved in. So this approach allows us to integrate the prior metabolic knowledge to improve the performance and also decrease the number of connections to make it sparse. So next layers are fully connected. And finally outputs the survival prediction of patients. So we trained 2, 3, and 4 layered architectures, versions of PyDeal. Okay, so in this one we discussed the experimental setup we used for evaluating our model. So we adopted a 5-fold cross-validation routine, as always, and we repeated 3 times with different Cs. That means 15 iterations in total. So we made sure that no patient and sample overlap between folds. And for a performance metric, we use time-dependent concordance index, or C index for short, which measures how a model is able to order the survival times of patients. Okay, so these are 3 baselines we use to evaluate our model against. The first one is Cox proportional hazard model, which is a linear model that uses proportional hazards function from the previous slide to measure the sample-dependent risk. The second one is random survival forests, which is a non-linear ensemble method, a very strong baseline too. And deep serve. So this model is fully connected feed-forward neural network that is introduced in 2018. It is only for survival analysis. So this is the performance comparison between PyDeal and 3 baseline models. As you can see, our model consistently outperforms all 3 baseline models in terms of median C index and improves over deep serve across 2, 3, and 4 layered architectures. So we checked whether the number of neurons in the first hidden layer affects the performance. So we trained deep serve models with 138 neurons in the first layer and compared. So it shows the performance comparison benchmark between PyDeal and deep serve with 138 neurons, and it outperforms 2, 3, and 4 layered deep serves again. And then we checked whether the regularization effect of our sparse architecture affects positively. So we again used deep serve with different dropout rates and compared to the our model PyDeal. So we can say that biology-induced sparsity here outperformed the dropout regularization in terms of C index metric. So to increase the interpretability and identify the most important metabolites for the survival outcome of PyDeal's predictions, we conducted downstream feature analysis via Shapley additive explanations model and calculate these sharp values for metabolites concentration vector on PyDeal's predictions. This is the calculated sharp values. So as you can see, alanine, glutamate, and glutamine were the top most important metabolites for our model's outcome. As I said, alanine, glutamine, and glutamate. So provide some context for these findings. We did a literature search for this and note that alanine was reported to be a biomarker in glioma in a recent study. Also, alterations in glutamate concentration have been observed in malignant gliomas. Finally, glutamine was reported as a glioma progression biomarker in multiple studies. So these findings show that our results are on par with the current literature. We continued with calculating the sharp values of pathway layers activation on PyDeal's output. And we revealed that mineral absorption, mTOR signaling pathway, and finally alanine, aspartate, glutamate metabolism were the most important pathways for PyDeal's output. So the last one was expected since alanine and glutamate were in the top most important ones. For the other two, we note that mTOR signaling pathway was reported before to have a key role in glioma metabolism. And calcium metabolism, which is connected to the mineral absorption, was found to be a glioma prevention mechanism. So again, our results resemble with the current literature here. So in this slide, we show successful predictions of PyDeal for rather unanticipated cases. So patient 253 lived more than 5,000 days after the surgery, and they had a malignant glioma. Our model predicted long survival for this one, which is a remarkable result since this patient was expected to have a much shorter survival time, unfortunately, but they did it. So, similarly, patient 350 with benign glioma only lived, unfortunately, for 42 days, and our model predicted low survival for this one. So, we can say that PyDeal's predictions in survival and prognosis is accurate and effective in this case. However, there is a limitation in our study. So our metabolite quantification models only consider 37 metabolites, which were manually selected by the pathologist. So 37 metabolites means 138 pathways in total. However, our metabolome consists of nearly 3,000 metabolites, and our consideration is only 1% of this information. So in a future study, we plan to expand these methods to include more metabolites, thus more pathways, to eventually improve the accuracy and, let's say, generalizability. Okay, so accurate feedback on predicted prognosis and hence survival can help the surgeon make more informed decisions on glioma patients, leading to better outcomes for these patients. And we show that biology-induced sparsity enables the training of deeper neural networks to also improve their performances. And finally, the increased interpretability on the importance of metabolites and pathways supports this surgical decision making. So thank you for your attention. Also, I want to thank the Doruk Cakmakci and Ercument Cicek for their support here. I would gladly answer your questions.
Thank you. It's a very nice study. Questions?
Thank you very much.
Thank you.
I had questions, comments on your results slide when you compare with the random forest and so on.
Yeah.
I mean, from what I see, deep serve is actually much worse than Cox on this data set, right?
The linear model, Cox.
Yeah.
Okay,
so deep serve, you showed it. It's worse than the one
No.
with the box plots, the previous one. It's really rubbish deep serve.
Yes.
And
Yes.
The other thing that I observe is that basically the number of layers doesn't make any difference.
Here, yes.
So have you tried just regressing the pathway level representation? Because the pathway level representation, I presume, is a non-linear transformation of the actual metabolites. So maybe you don't need any further metabolites and you can interpret beautifully without any sharp stuff.
Okay, yeah. So thank you for this question. So for Cox and deep serve.
Well, that's not your problem, but
it's
just a meta problem that,
Yeah. So for problem in deep serve is that deep serve is a fully connected neural network, and we have a data set of size 400. So when we increase the number of layers, it drops. You see the median C index drops. However, in our model, as we induce sparsity here, biology-induced sparsity, the median C index just not marginally increases.
You don't have any sparsity afterwards.
Afterwards, no.
Yeah,
so maybe the first layer induced sparsity is better for. Yeah.
Maybe it is representation of the level of pathways.
Exactly.
Just regress that.
Yes, I will try that. Thank you.
I have one question. So in the gliomas, that IDH1 mutation is also very commonly seen. So
Yes.
So that enzyme, IDH1 is actually an enzyme. So that's also linked to the glutamine. So they are kind of interlinked with it. So the samples that you are using, do you have the information of which ones are IDH1 mutant?
No.
Or can you integrate that information as well to your model?
Actually, we don't have it. We have only the HRMS output and the survival times or the disease or not disease information. However, as you said IDH induces malignant gliomas to have more survival times. So we do not consider this explicitly. However, maybe our model sees some metabolic profiles and understand that this patient has IDH mutation maybe, so it predicts high survival for these ones.
So if you have the samples, if you want to just sequence them just for the IDH1, so that can be maybe extra layer of input. For
Yes,
the model.
exactly. Thank you. Thank you.
What's the number of dimensions, the input dimension for the network?
Okay, so for raw output of HRMAS, it is 60,000 elements, one spectrum. But we decreased this to 37 metabolites.
I was describing that maybe the Cox proportional hybrid problem, that poor performance would be for the high dimension.
Yes, yes. Well,
so you can use, for example, penalize the.
well, we did a grid search on hyperparameter selection on Cox pH. So probably yes, yes. It's breach or
A bit of a naive question, potentially outside of the scope, but I don't quite see how the prediction actually will affect the decision of the surgeon.
decision of the surgery. So
Can you
yeah,
comment
this is what our collaborators at University Hospital Strasbourg asked from us. So I haven't thought of this before, but maybe they decide on whether to operate on a patient or not. Maybe the survival time.
But
you
it
need
happens
data from
during
the
the
patient.
surgery. isn't it?
Yeah, maybe the further resection can happen, or they can stop resecting tissue.
They keep chopping it.
Maybe they excise more.
More or less maybe.
Yes.
Like a
Yes.
pre-informative.
Because our prior work on this feedback mechanism included surgeons to pick some spots from the excision cavity and then take those predictions of these tumor samples from the resected tissue, so they can know how much tumor is there left. There.
So they normally send like immediately those removed samples to the pathology and they do like a fresh frozen and dissection and then stain it and they immediately report back to the surgery room. So there is a kind of room for waiting at least half an hour to 1 hour.
Yes.
And the skull has been opened. So that's
Yes.
what I heard.
And usually the manual feedback, they need to have a pathologist and an NMR technician and maybe or something surgeon with histopathological information needed in the surgery room. But with this one, we don't have any need. So maybe considering the surgical time constraints, this is very quick and rapid.
I think there is recently there is a trial to nanopore sequence, a shallow nanopore sequence, and see the methylation and classify the subtypes of brain tumor. How do you think your method compare with those kind of approach?
So you are asking that for each has histopathological. Can you ask again?
So recently, during the surgery, so some group try to do nanopore sequencing, shallow nanopore sequencing, and see the maturation and do the classification of the subtypes of brain tumor,
Okay.
and then determine the option of surgery. So how do your method compare with those approach?
Okay. So our prior work on feedback mechanism again included this histopathological tumor classification. So this is on top of it. We will have that in tumor surgery room and this novel one. So we of course compared it with the other classification ones, but for glioma ones, this is the state of the art for classification and survival analysis.
Okay, thank you. If you have more questions, we have a poster session. So thank you for a nice talk.
Thank you.
So,